My name is Sara Colpitts. I am a postdoc in the lab of Leo Lefrancois at the University of Connecticut Health Center. I graduated from the University of Connecticut with a bachelor's and a master's in 2003. I then went to the University of Pennsylvania and I did a PhD in immunology, which I finished in February 2009, and I've been here ever since. So I've been here just about over 4 years now. I work on a protein that's called Interleukin 15. We abbreviate it, we call it IL-15. And at least from an immunological point of view, IL-15 is important because it's what we call a survival cytokine. It keeps cells alive. And two of the cell types that are really dependent on IL-15 are some CD8 T cells and NK cells. And they are really important in fighting viruses and also tumors. So those are kind of cells you want to have around. And I don't actually work on those cells themselves, but I work on the cells that make IL-15. And we've done some experiments. We sort of started this project by looking at IL-15 in vivo. And I should mention that all the studies that I do are in mice. So everything's sort of preclinical. And we looked at how IL-15 expression can change with inflammation, and the inflammation we chose was a virus infection. And then looked at some of the inflammatory pathways that were required in order to mediate these changes in IL-15. And so that work got published about a year ago. And lately, the two projects I've been working on, well, actually, I'm hoping to submit a paper tomorrow that's looking at some novel sources of IL-15 in vivo. And then yesterday, I was doing a pretty big experiment where we have a collaboration with some people in Texas and to look at the role of some of the microbiota that's present inside the gut and how that might be influencing IL-15 production by some of the cells in the intestine.
Can you speak about how IL-15 expression changes following viral infection?
Sure.
And how those changes help you identify which inflammatory changes are necessary for upregulation?
Yep. So a lot of what's been done with IL-15 up until, about 4 years ago when I was starting this project, it's been done in vitro. And so it had been shown that if you can grow cells in vitro in culture, and if you throw different inflammatory stimuli on them, they will upregulate their IL-15. So we had pretty good candidates as to what we thought was going to mediate changes. But what was unique about the studies we were doing is that we were doing them in vivo, in like a whole mouse, rather than in a culture system. And so we went with a viral model, and we chose a model called vesicular stomatitis virus, which is probably more deadly to a mouse than a human, but and pretty easy. We infected the mice with VSV, and we can show in vivo that the IL-15 is upregulated pretty dramatically as soon as 24 hours. And it comes up really quick, and then it goes back down really quick. And since we're working in mice here, we were able to use some genetically deficient mice in two of the more prominent inflammatory pathways. There's a bunch of them, and the one we ended up seeing an interesting result with was called the type 1 interferon pathway. And what happens is if you get rid of the receptor for the type 1 interferons, the cells completely lack the ability to upregulate IL-15. And we kind of did some cool, what are called chimeras, where in the same mouse you have normal cells and cells that lack the interferon receptor. And so even though they're experiencing the same inflammatory environment, the normal cells are able to upregulate while the deficient cells don't even change their expression at all. So your immune system is really divided into two big arms. You have your adaptive arm and your innate arm. And the main difference is that the adaptive arm is specific and the innate arm is nonspecific. And IL-15 is produced by cells in the innate arm. And the specific cell type that most people study is called a dendritic cell, or we abbreviate it calling it a DC. DCs happen to be easy to generate in culture, so it's kind of, 20 years ago or 15 years ago when we were limited in some of the technologies, you did what you could. And so you could grow DCs in culture, And then you look for IL-15 and oh great, they make IL-15. So that became a pretty good system for studying cells that make IL-15.